This is an overview of an article entitled "A Polygenic Risk Score for Coronary Heart Disease Modifies the Elevated Risk by Cigarette Smoking for Disease Incidence," published in the January 2018 issue of Circulation, Genomic and Precision Medicine. It can't be emphasized enough that cigarette smoking is the #1 single strongest modifiable risk factor for coronary heart disease, or CHD. There are many good reasons to stop smoking, but one of the best reasons is the fact that after a person stops smoking, the risk of CHD diverges from that of people who continue to smoke and approaches the risk of people who never smoked. Even though we know that smoking is a powerful risk factor for CHD, and that one's genetics also can affect risk of CHD, a relatively unexplored question is whether smoking affects genetic risk of CHD. The authors of the study under discussion set out to answer this question. They used a polygenic risk score comprising 50 common variants previously found to be associated with CHD in genome-wide association studies. The score provides a way of assessing the aggregate effects of a number of genes on disease. The authors calculated polygenic risk scores for more than 24,000 participants in the Malmo diet and cancer study in Sweden, who had been followed for many years. About 13% of these participants had incident coronary heart disease events during the time they were being followed. The individuals in the study were stratified by smoking status: never smokers, former smokers, and current smokers, the last one defined as having smoked in the past year. Here are multivariable adjusted odds ratios for coronary heart disease. Not surprisingly, the higher the genetic risk score, the higher the risk of disease. Current smokers had substantially higher CHD risk than never smokers, with an odds ratio of 1.67. In contrast, former smokers were much more similar to never smokers, with an odds ratio of 1.19. It should be noted that these observations are very consistent with previously published studies. Here's the answer to the major question posed by this particular study: whether there is an interaction between smoking status and genetic risk. Here you see data for subgroups defined by PRS, polygenic risk score, tertile, low, medium, and high, and by smoking status. The first notable observation is that among people in the lowest PRS tertile, current smokers had double the risk of never smokers, with former smokers being intermediate. This contrasts with people in the highest PRS tertile, for whom smoking had less of an effect on CHD risk. The second notable observation is that among never smokers, indicated in blue, a high polygenic risk score predicted double the risk of that seen with a low polygenic risk score. This contrasts with current smokers, indicated in red, for whom the PRS had less of an effect on CHD risk. Perhaps the key point to take away here is that smokers with a low PRS, or to put it another way, people with, quote, good genes, unquote, have almost exactly the same level of CHD risk as never smokers with a high PRS with, quote, bad genes, unquote. In this context, smoking does as much harm as having bad genes. Formal statistical analysis found that there is indeed a significant interaction between smoking status and polygenic risk score. Notably, the interaction was stronger in men than in women, although that might in part be due to the fact that there were fewer women in the cohort with incident coronary heart disease, and so the women-only analysis was underpowered. Interestingly, the smoking PRS interaction was unchanged after adjustment for family history of heart disease. It's also worth noting that individual variants near the genes PPAP2B and HDAC9 had the strongest interactions with smoking status with respect to CHD risk, although whether these interactions are genuine and the mechanisms by which they might occur will require follow-up studies. In conclusion, this study confirmed that both a polygenic risk score and smoking status were associated with coronary heart disease risk, and the study found a significant interaction between the two risk factors. The polygenic risk score did not seem to have as much of an effect on CHD risk in current smokers, perhaps because their risk was already substantially higher due to their tobacco use. As such, one of the key implications of this work is that smoking largely negates the protective effect of a low PRS score, or to put it another way, the protective effect of good genes, highlighting the fact that the choices we make in life can override the effects of the genes we inherited from our parents.